So in this video, we're going to talk about how a phase contrast microscope works. So in the last video, we saw how light which passes through cells can become retarded relative to the light which passes through the media only around those cells. That's typically gets delayed by phi equals pi over 2. So we're going to learn how now a phase contrast is able to convert those changes in phase, which are normally invisible, into differences in intensity you can visualize at the imaging plane. It does so using something called interference. So if these two waves are in phase, then at the imaging plane, they'll combine together and basically superimpose on one another. In this case, wherever you have a peak, you have another peak, so at the imaging plane, you end up with a much larger peak. However, if these two waves are out of phase with one another, that is, phi is equal to pi in this case, wherever you have a trough, you have a peak, so when you ever add these two together, they'll basically cancel each other out at the imaging plane. And we saw before that the amplitude at the imaging plane, the intensity of the imaging plane is proportional to the amplitude squared. So, and if this is the case, if this occurs, then you'll end up with regions with very low intensity. So it's important to note these are two limiting cases. There's phi is basically a continuous variable. It can go, it can take on any number, and so these are just two endpoints. So you can have varying degrees of constructive or destructive interference depending on the actual phase difference between these two waves. It's also important to note that if phi is, for example, 2 pi, then these will be out of phase with one another by exactly one period. So again, you'll get constructive interference. So how does a phase contrast microscope work? Well, you take a standard bright field microscope and you add 2 bits of equipment to it, which will turn it into a phase contrast microscope. The first thing you need is a condenser annulus. So here's your light source here shown in yellow. It basically passes through this condenser annulus, which blocks all the light passing through the microscope except for the region shown here in white. So you end up with a hollow cone of light passing through your specimen and going to the other side. So we saw in the previous lecture how when light passes through your specimen, how it diffracts, and it moves into different regions of your optical path relative to light, which simply passes through unperturbed. So we're going to consider 2 different regions of light here. We have the light which is diffracted by your sample here, shown in red, and the surround light here, shown in yellow. That will strike the second piece of equipment you add to your bright field microscope to make it a phase contrast microscope, which is called the phase plate. Now the phase plate does 2 different things. First off, it can tune the difference in phase between this surround light and this diffracted light by having different thicknesses. So your surround light will pass through this region here in a positive phase plate here, which is thinner than the region which your diffused light passes through. We know that after it exits the sample, your light which is passed through a cell is already retarded by approximately pi over 2 relative to that of the light which passed through the media and is passed through unaffected. This phase plate will basically retard this by another pi over 2, so you can get a full pi difference in the phase between these 2 different waveforms here, allow you to achieve destructive interference of the imaging plane. And it does so by basically in a similar way which we derived in the previous lecture. It's thinner, and this has an index of refraction which is greater than 1, so the light which is passing through this thinner region here gets slowed down less than the light which is passing through this thicker region here. Now the second thing that this phase plate does is it damps the amplitude of the surround light. So you know from the previous lecture last week that the amount of diffracted light which is induced by your sample, which is basically caused by moving through your cells, is relatively weak compared to your surround light. So your surround light is quite a bit more intense, and you need to get these 2 different, your surround light and your diffracted light, in such that they have a similar amplitude, so they can fully destructively interfere with one another. And to achieve this, they put a partially reflective metal coating here in this ring-shaped region, which will reduce the amount of light passing through by 70 to 90%, which will basically cut this down and your surround light intensity down so that it's a comparable level to your diffracted light. So we have two different types of phase contrast. We'll first talk about, we've been talking so far about positive phase contrast, and that's when you can basically this phase plate shifts your diffracted light by another pi/2, such that you get destructive interference occurring at the imaging plane, and that allows these two different, the surround light and the diffractive light, to cancel each other out. So wherever you have cells, you have this pi phase difference, which leads to a smaller intensity in the center of these cells. So cells appear dark relative to that of the background. This is our red blood cells. You can also look at thicker samples like a termite claw. So these red blood cells on their own, they induce about a pi/2 phase shift. However, in these thick samples, on the edges of these samples, you can see that you also get this destructive interference. This phase plate has caused a full destructive interference to occur, a full phi equals pi phase difference at the imaging plane, and you get again this dark region here at the edge of this sample. Now there's another type of phase contrast which is used more rarely than positive phase contrast, and this is known as negative phase contrast. So again, you have a darkened ring-shaped structure in the middle of your faceplate, but now that ring-shaped structure in your faceplate is thicker than the region where your diffuse light passes through. So this will essentially undo the pi/2 phase shift induced by your sample, so that the phase difference between your surround light and your diffracted light is now eliminated, and you can get constructive interference at the imaging plane. So in this case, your diffracted light and your surround light are now in phase with one another, and the imaging plane, they combine with each other to give you a region with higher intensity wherever their cells are present. So now cells appear white rather than black. And in your thicker termite claw, you can see that these thin edges of this termite claw, you again get you again get constructive interference, and you get a bright region along the edge. Now, the way you achieve, the way you build a phase contrast microscope is you typically have a special turret that sits in your condenser, which basically gives you this hollow cone of light. So in the bright field position, this is typically left open. You let all the light through. Whenever you use phase contrast, you can rotate this into place, with this annulus into place, which only lets through this hollow cone of light. You can also put in the prisms, which we'll talk about in a second, for DIC into the same rotating turret. Now, each different magnification of objective you use in phase contrast requires a different annulus. So this is the phase plate that's in different magnifications of objectives. It's this bit here in the middle. You can see this is a different size, so you have to have the corresponding annulus which matches this. And we'll see in a second how these must be aligned with one another. So this phase plate is typically built into the objective, whereas the annulus, you can adjust that separately by rotating this turret up top. So to get a good phase contrast image, your phase plate and annulus have to be well aligned or perfectly aligned with one another. So this is a case when you get these two are in alignment. If one shifted relative to the other one, however, you end up with a rather poor bright field image. Now you can adjust the position of these with what's called a Bertrand lens, which will allow you to observe both your annulus and your phase plate through your eyepiece. And you can use small thumb screws to basically move your annulus into the right position, so that you achieve good overlap between them. Now, there's obviously some drawbacks of phase contrast, and one of these is it tends to induce artifacts, which can be quite distracting depending on your sample. So this is caused, so if you look at these cells here, we'll look at exactly what it looks like first. So you can see around these red blood cells that you tend to get this halo here, which is not real, which is induced by this phase contrast technique. You can also see this halo around the termite claw. This is caused by the fact that you cannot completely separate this diffracted light with the surround light. Some of this diffracted light actually passes through this darkened region of the phase ring, which induces a phase shift in it. So some part of your diffracted light is basically phase shifted, like your surround light is by your phase plate, and that's what generates this artifact around cells. So you have the halo artifact we saw around the red blood cells. In larger objects, you can see what's called a shade-off artifact. So the center of this appears a bit brighter than the edges, and this is caused by a similar problem in the optical path. You can also see this in negative phase contrast as well. So when it's working correctly, phase contrast can definitely give you a boost in signal. So this is a bright field image, a centered bright field image. This is a phase contrast image of the same sample. You can see these. You can observe that you can visualize these nuclei quite a bit better in phase contrast rather than standard bright field. The difference is perhaps even more apparent in this brain tissue, where you can see these cells quite a bit more clearly. You have a much larger contrast, as we defined in the last lecture, compared with a bright field microscope. The other potential problem of phase contrast is that we often want to take a fluorescent image at the same time as we take a transmitted light image. And there, the fluorescent light passes up through your objective and to reach your specimen, which then can emit back your fluorescent light back to your imaging plane. Now the problem with this is light has to pass through your objective. And if you use a phase contrast objective, it has this phase plate in it, which has this darkened ring in the middle, which can reduces the intensity of light which passes through that ring by 70 to 90%. It's not all the light passes through this region, but the bit that does gets damped down quite a bit. And this can reduce the amount of fluorescent signal you get through your system, so it's obviously not as good as a bright field microscope or a bright field objective for fluorescent imaging. You can't remove the phase plate from a phase contrast objective. It's basically built into it. And that can be an important consideration when you're trying to consider what kind of transmitted light image to take along with your fluorescent image. Another big advantage of using phase contrast is that it helps you delineate cells with no closely packed together. This is some experiments from my lab that shows Cinnamonas aeruginosa moving around collectively on a glass surface. So each one of these are individual cells, and one thing we like to do is try to build automated cell tracking routines to follow the movement of individual cells to analyze their behavior. Now these are very difficult images to analyze because these cells are closely packed together, and if you try to train a computer to basically detect individual cells, you run into a problem in what's called segmentation. So you have two cells which are close together. Get my hands in the right place. In one of these frames, they can potentially overlap with one another, which will trick the computer into thinking that there's actually one cell when there's in fact two. So when cells overlap and move apart, then that really impacts your ability to track individual cells, because one of those cells will disappear whenever this happens. So phase contrast will it creates these halo artifacts, which are, in this case, we're using positive phase contrast, these are relatively low, and gives you these halo artifacts which are lower, which are brighter around the cells. But that actually helps you out, these artifacts, in this case, because it boosts the contrast. It gives you a cleaner edge to these cells, which you can use something called watershed to basically delineate the edges of individual cells, which then you can subtract from your original images to basically better distinguish these individual cells and basically draw a line in between them where two cells are touching to allow you to segment them properly. So if you do this the right way, you can use this increased contrast, which is afforded by phase contrast, to actually track individual cells when they're collectively moving. And here we've basically trained this machine learning based algorithm to follow individual cells when they're collectively moving. Some of these fields of view have up to 10,000 cells, and we can track each one of them in a single image or in a single time course. So here we're plotting the centroid of each cell with this little blue dot, and these orange trajectories are following the path of these cells as they move through time and space. Now, there's another form of phase contrast microscopy which is a bit more advanced. And this is called quantitative phase contrast microscopy. And in this case, you don't use one phase ring, you use many different phase rings, which can basically adjust the phase difference between your diffracted light and your surround light relative to each other. So these basically shows different phase rings where you have no phase ring in this case, or a phase ring which introduces a no phase shift to ones which induce a larger phase shift here. And this causes the light, your surround light and your diffracted light, to constructively and destructively interfere with each other in different ways for each one of these different phase rings. And this will allow you to use a model of interference to basically work out the optical path length at different points in your sample. And your optical path length is simply the thickness of your sample times the refractive index. So you can see how that product will change laterally across your sample. So this is a neuron. You can see how the optical path length has changed in different locations. Now, in a biological sample, you can't necessarily tell differences in thickness of the sample versus changes in the refractive index, since you can only measure the product of these two quantities. But if you have an object which has the same refractive index throughout it, then this technique will allow you to measure very small changes in thickness across the width of your sample, because you're simply you're basically using the wavelength of light as a ruler to measure these changes in thickness. So this can be a powerful approach, and you'll read more about this in the session later on this week of how this has been applied to biological samples.